We cannot hear anything. It's okay, Miguel?
Oh, you cannot hear?
No.
Let me check what's going on. I think it's in the share settings. Yeah, this, okay. Share sound. Maybe you can, let's see. No?
Yes. Now
Okay,
it's
okay.
good.
So let's start again. Sorry. Okay. So everything good with the audio now?
Yes, perfect.
All right.
Good.
Awesome. So welcome again to our first session of the first day of our club in Science Club International, building the synthetic cell from bacteria to stem cells. Today I will go through a recap of cellular biology. I think for you folks, some of the concepts might be, some of them might be redundant or you already learned them in your college program. But we want to bring everyone up to speed, and in the next sessions we'll cover more, maybe more advanced content. We live in an exciting era full of exciting discoveries in synthetic biology. You can see this example of 2010 when some of the first DNA encoded computers started to be possible. And who knows, maybe in the future we'll have some forms of encoding information in DNA. And this might have important applications in nanotechnology or areas where we need more and more miniaturization and more efficient use of batteries. There is also 2012 advances in Symbio. One of them is the plant-derived artemisinin that has been switched to a method in yeast. So normally in this top panel, you can see the cultivation of the plant that creates this drug artemisinin. Takes a long time. It's almost 14 to 18 months, but the extraction methods are very expensive and they won't produce a lot of the drug, which is used to treat forms of plasmodium infections and parasite. It's an anti-parasites, but during this last decade, there has been methods to use yeast and sugar to create one of the precursors of the drug, and through some small chemical modifications, you can have artemisinin. And this is called synthetic artemisinin and has been one of the leading most sought after drugs. And again, synthetic biology is putting the gene of this plant inside yeast. There is this other example, more recent, for T-cell therapies. So you can see here a T-cell that recognizes a cancer cell. And in response to the cancer cell, it will start producing cytokines, such as IL-12. You can customize this response to produce maybe cytotoxic agents or adjuvants to differentiate the T-cell into a different type of T-cell through the expression of transcription factors, or to produce therapeutic antibodies. So you can essentially tell the T-cell what to do when they respond to a certain antigen. And this will be part of the future and the next generation of cancer immunotherapies. But to understand all these advancements, we want to start understanding what is a cell, how does it transcribe their genes, how does it store the information, and also what's their spatial location. So we are talking about, in a finger, there is this width of 20 millimeters, but if you go just a tenth of that, you can start seeing this structure in the finger. And going even lower, you can see the cells in that part. And they can see the skin cells, maybe some stem cells that renew the skin all the time, and the fibroblasts. And so there's the dermis and the epidermis in those fingertips. If you go even lower, there will be the mitochondria of the cells. Here, you can see the organelles like the endoplasmic reticulum, the Golgi apparatus, the nucleus, at the scale of 20 micrometers. And this is how you will look them in a microscope, mostly at this resolution, 0.2 millimeters. There might be some powerful microscopes that give you at this scale. An electron microscopy could give you resolutions for the mitochondria individually. There is also known what is inside of the mitochondria. And here, when we go down to proteins, like this one, in the scale of 20 nanometers, and molecules, which are 2 nanometers, this is the scale of the DNA, and then atoms, which are at 0.2 nanometers. Well, these are molecules, actually, but individual molecules. So this is to give you an idea of what the scales are, and we will be talking today about the scale of 2 nanometers for DNA. We'll also be covering some of the ribosome and proteins at the 20 nanometer, and we'll show some images of cells which are in those previous scale. Just quickly to summarize the components of a cell, as is classically known, it's the nucleus. The nucleus has this structure called the nuclear pore that will control the intake and and the transport of molecules inside and outside the nucleus. There is also the chromatin, which will have the DNA inside the nucleus. Then there is the ribosomes that make proteins. There is a rough endoplasmic reticulum that harvests a lot of the ribosomes, and that's how the proteins will assemble. And then some of the proteins can get modified in the Golgi apparatus or other structures. The Golgi apparatus also will be in charge of lipid synthesis. And there is the mitochondrion, which is the powerhouse of the cell, will have a lot of the processes that give energy in the cell and produce ATP. There is the lysosome that get rid of many of the toxic or non-use components in the cells. And the cell membrane made of a bilipid layer that protects everything inside. But it's also allowing the interaction through transporters and also through some forms of diffusion. So this is the cell. We're going to start talking about inside the cell, which is the DNA and chromatin, as these modifications that in synthetic biology are made will be targeted at the DNA level to change the rest of the behavior of the cell. So fibroblasts, myoblasts, or nerve cells under the microscope are looking like this. This is an optical microscope. You can see they look all very different. That means they have different ways to regulate their genes, and we'll go back to this concept. But as you can see, again, the scales are 20 microns, which are the size of those cells. So, okay, so going into DNA. DNA is inside the nucleus. It's made by 4 types of nucleotides. And these nucleotides are guanine, cytosine, adenine, and thymine. They will be joined together by a phosphate bond, phosphodiester bond that connects each of them from the phosphate in the sugar backbone. Okay, so the base is actually a chemical base. It has has amine groups and can be donating electrons, but that's the definition of a base. But when we have it together with a sugar phosphate and well, with a sugar and a phosphate, that makes a nucleotide. This structure is called nucleotide, and the nucleotides assemble each other from the sugar. Okay, so the bases are not really connected chemically. It's the covalent bonds between the phosphate on the sugar backbone. So this is a more close view at the nitrogenous base, which has nitrogen. It's a chemical name for a base, which has the amine groups. Then it's joined to the sugar, which in the sugar we name the carbons from 1 to 5 prime. And the prime is just a sign that is used. It doesn't mean much. It's just 1, 2, 3, 4, and 5 in the here. So yes, and what we can see then is that once the phosphates are joined together, there will be hydrogen bonds between the bases of 2 strands. So what will happen is that there will be a single strand of DNA that is joined to another strand in this double helix structure. And then the bases themselves will be joined by hydrogen bonds on both strands. Okay, so that's kind of what is known of how DNA is made. What encodes for information is the order of the bases in the strand. So depending on where is adenine, thymine, and what goes next, that's encoding for information. Okay, but once DNA is inside the nucleus, it has to be very tightly packaged. So you might have heard that a strand of DNA from one cell, if we extend it, it will be 2 meters long. But it's packaged. It's all packaged inside one single nucleus, and for that it has to go through all this process of DNA wrapping around histones, which are proteins. These histones will have a positive charge, and the DNA has a negative charge because of the phosphates. So it will join by electrostatic interactions with the histones and then make a chromatin fiber. And then through multiple rearrangements, some of them are not well understood, it will create condensates. And finally, the chromosomes, which are the most condensed form of DNA. So all the human genome is packaged in 46 chromosomes. Here, this picture is colorful because it's using a method called FISH, and this is called fluorescence in situ hybridization. So with this technique, you can actually take all the chromosomes inside a cell and then use a fluorescent probe that has complementary regions to each chromosome, and they have different fluorophores. So it's a way to visualize the chromosomes, knowing the sequence of the chromosomes and sequence that are specific for each of them. And then once you arrange them artificially, this is how they look on the left from a lysed nucleus. But once you arrange them artificially, you can call this a karyotype. And you can see that there are 2 of each chromosome. This is chromosome 1 because it's labeled in yellow with that probe. And then there is chromosome 2, which is labeled in red, and so on. So we have 2 copies of each chromosome. That ensures that we have a backup copy of every gene. And therefore, if there are errors in one of the genes, we have a way to repair those errors. And there are mechanisms that can use a good copy to repair a bad copy. So this is a very important part of evolution. And for the woman, there will be 2 X chromosomes. And for man, it would be X and Y. So that's how it would be. Now, in women, there will be also silencing of one of the X chromosomes by long non-coding RNAs, which we will talk again later. So the genetic information is carried in DNA, okay, and it's expressed as functional products. So from gene A, you will have protein A, gene B will code for its own protein, and gene C its own protein. Like now, not all of the genes will code for proteins. It's only 2.5% of the genome that encodes for proteins. But this is what is called, what is known as the central dogma, where one gene goes transcribed and then translated into this direction. Okay, so the first part is transcription. So DNA in the double strand that we have seen before is joined by the backbones. It's coming from 5' to 3' in one strand, and in the other direction in the complementary strand. Sometimes one of the strands is called template, and the other will be the coding. And then through transcription, one of the strands will be converted into RNA. The difference between DNA and RNA is chemically that the sugar has an oxygen in the RNA. It's an extra oxygen that is not present in DNA. And this oxygen actually makes a big change because the oxygen makes RNA less stable. DNA is very stable because it doesn't have that oxygen. But RNA also has a different base. Instead of thymine, it will have uracil. So it's slightly different, but it's a very similar compound in general. And it's like its cousin of DNA. But RNA is a single strand, and it will be the first step in gene expression, okay, from DNA to RNA. So we'll watch this. After this, we'll watch a video, and then we'll have a break, a summary, and a break. So this is just the last slide where the polymerase is a protein that will be key during transcription. So what the polymerase is doing is going to bind DNA, and then it has a hole where there is uptake of ribonucleoside triphosphate. So all the components of RNA individually will go inside this hole. They are called nucleosides because they are not yet with one single phosphate and in the chain. So They have this different name. There is also magnesium ions that are required to make the synthesis. And as you can see, the DNA is unwrapped at the section. It's like a bubble where the transcription is going in one direction. This goes really fast, and it will happen all the time in the cells because they need to express different genes. So we're going to go to the video, and then we'll go to the questions, okay?
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Miguel, we cannot hear you.
I'm sorry. Yeah, I will read the recap in English, and then maybe some of the volunteers can read in the other language. So the past decade has been full of discoveries in synthetic biology. To understand them, we started looking at the organelles in the cells. We also reviewed the structure of DNA and its organization into chromosomes. Finally, we explored how RNA polymerases transcribe information coded in DNA.